we start with the basics. So we start down there with the molecules, and then we're going to build our way up. Now, what I've done here in these first few sections is I've kind of broken this down into the bits from your past science classes that I really want you to know and remember, okay, because we're going to build on those concepts. So unfortunately, you've taken, well, not unfortunately, but you've taken a lot of science. Not all of it is going to directly apply. Particularly in phys, there are key aspects of some of the disciplines you've explored, particularly chemistry and a little bit of molecular biology, that is going to play a role in your understanding of how the human works. All right, so I try to start my talks with objectives. The idea behind objectives, they're not just for me, they're for you too, okay? In each class that you have, the objectives for each session are what you should be taking away from that, okay? So you can actually use the objectives as a kind of study guide, all right? If by the end, when you're studying for the exam, you should be able to fairly completely do whatever the objectives tell you to do, right? So it's just kind of a guide. So we're going to talk about diffusion and osmosis. We're going to talk about water. We're going to talk a little bit about the major players molecularly, carbohydrates, proteins, lipids, and nucleic acids. We'll talk a little bit about the energy currency, adenosine, and then we'll talk just a hair about enzymes to refresh your memory from biochem. All right, so osmosis and diffusion. Osmosis and diffusion are both about motion. Technically, they are the same thing, okay? Diffusion is inherent to the universe. Because molecules are always wiggling around due to their thermal energy, it means that things are always drifting, okay? Molecules are always moving from areas of high concentration to areas of low concentration. The only time this doesn't occur is at absolute zero, right? So you're talking about intergalactic space. That's the only place this doesn't occur. Everywhere else it occurs, and it occurs to a greater extent the higher the temperature is. So we human beings, we walk around at 98.6 degrees Fahrenheit. That's a fair amount of thermal energy that's sitting there driving diffusion all the time. So we have this Brownian heat motion that causes molecules to move. The key is the direction. High concentration to low concentration, all right? That is the direction of motion that is free, okay? The universe powers that. You don't have to put any ATP in. You don't have to put any heat energy in. You don't have to put anything in. Diffusion's going to occur automatically, okay? So as long as there's a difference in concentration between one region and another, and the molecule can physically get from the high concentration area to the low concentration area, diffusion is going to occur. That molecule is going to move. Now, diffusion will continue until the concentration gradient disappears. All right? So a classic example is, you all know what mothballs are, right? If I took a bucket of mothballs, set it here on the floor, and opened the lid, eventually you'd be able to smell the mothballs all the way in the corner of the room, right? But not right away. That's diffusion. You got a high area of concentration over here, low area over there. Eventually the mothball smell will drift until the concentration gradient disappears, right? So why is this basic thing so important? Diffusion powers the movement of most important molecules in the body, okay? And the reason that we evolved to utilize this is because diffusion is free. We don't have to pay the energy dollar to move things by diffusion, all right? So diffusion is the cornerstone of how things are separated and move in the body. So we'll be talking a great deal about it, particularly in our next chapter when we start building a cell and building a membrane potential. Okay. Now, osmosis is the movement of water from areas of low solute concentration to areas of high solute concentration. Okay, so in a solution like water, you have your solvent and you have your solute, right? So you have two different substances that are mixed together. Well, osmosis is talking about the movement of the solvent, of the water, separate from the solute or what is dissolved in it. All right? So the way I think of it is osmosis moves opposite of diffusion if you're talking about the solutes. Okay? So in a solution that has a high concentration of solute, right? Diffusion is going to move that solute from areas of high concentration to areas of low concentration. That's diffusion, right? Osmosis is going to move the water from areas of low solute concentration to areas of high solute concentration, all right? So some more in pictures here in a minute. Now, some of you, depending on your chemistry teacher, may have learned of osmosis as the diffusion of water. Did anybody learn it that way? A few of you did. Okay. Also true. Osmosis, another way of describing it, is water moves from areas of high concentration to areas of low concentration. But because we don't typically think of water's concentration, right? We think of the solute. I use this system instead. So we go from water moves from areas of low solute concentration to move to areas of high. Okay, so diffusion in 4 pictures, right? Because molecules are always wiggling around, over time, if we have an area of high concentration over here, area of low concentration over here, right? Over time, wiggle, 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 our concentration gradient disappears because everything has spread out. Right? The high has gone lower, the lower has gone higher. All right. Now, what about osmosis? Here in our picture, blue are water molecules, purple are other molecules. In this case, it's sugar, right? So what we have here, we have a semi-permeable membrane. Okay, what does that word mean? A semi-permeable membrane allows some substances to pass while restricting the flow of other substances. Okay, that's all semi-permeable means. Now, typically, a semi-permeable membrane that we talk about in human phys is permeable to water, but not solute. Okay? Now, why do we do that? Because that's our cell membrane. That's why. The cell membrane is permeable, well, almost everywhere in the body, not the kidney, but almost everywhere. The cell membrane is permeable to water, but only selectively permeable to solute. Okay, that's why we talk about osmosis, because all over the body, we got semi-permeable membranes. Okay, so in the case of semi-permeable membrane concentration difference, everybody sees that, right? What we're going to see is that water moves from areas of low concentration to move areas of high concentration. The solute can't move, so the water moves instead until the concentration is about the same. Now, you'll see that we have actually more water on this side now. That's what we call osmotic pressure. Osmosis will continue to push water until one of two things happens. Either the pressure difference becomes great enough, so the column of this water is heavy enough that it can't move anymore because it's pushing down, right? Or when the concentration gradient on the two sides becomes equal, right? Okay. So, who's learned about osmosis before? That better be everybody. Come on. Everybody's learned about osmosis. Now, the confusing bit, I think, about osmosis is this part, that what we can have is we can actually develop a physical pressure from a concentration difference. Now, where do we see this happen in the body? We see it happen in the kidney, all right? In particular, in the concentration system, where we take very dilute urine and we make much more concentrated urine out of it. Okay. All right, so I think I talked about all that. Yep. Whoop. There. Yeah. Okay, so that's diffusion and osmosis. That's how things move. Now, what moves around? Well, that's going to be our biologic molecules. I swiped this from a different book because it's nice and simple. You know, some kid at some point in your medical career is going to ask you what a human being is made of, right? Well, here, this is what a human being is made of if you want to give them a technical answer. We're a lot of oxygen and a lot of carbon and hydrogen. Okay, why? Because we're mostly water. That's the hydrogen and oxygen, right? Carbon we have a lot of just because it's simply in everything, okay? So in the parentheses there is percent of total body weight. So, you know, now you can go tell everybody you're 18.6% carbon, right? Okay, oxygen, carbon, hydrogen, nitrogen, those are the big players. Nitrogen, because it's found in most compounds, number one, and in our bodies, proteins, right? Proteins contain nitrogen, and we have a substantial amount of protein in the body, therefore we have a substantial amount of nitrogen. Now, what about the others? We have a fair bit of calcium. One of the things you're going to hear me say multiple times this semester is calcium is my favorite ion, okay? And it's because in the body, calcium is what makes all the cool stuff happen. Calcium makes action potentials happen. Calcium makes muscles contract. Calcium even makes secretion occur. So calcium is awesome. Well, the 1.8% of total body weight of calcium, though, where's most of that? Yeah, it's in your bones. We actually only have a tiny little amount of circulating calcium, but that tiny little amount is critical. You'd be dead without it, right? So the cool thing about calcium is we store a lot of it because it's hard. You know, calcium is hard and durable, so we make bones out of it. But it also means that this precious ion calcium, we walk around with a storage of it all the time. So we never run out, so to speak. So that's calcium. Phosphorus goes along with calcium. The crystal that forms bone is mostly calcium, phosphorus, and then organic compounds like collagen. All right, and you can imagine that your skeleton, you know, weighs probably about 10% of your body weight. So calcium, phosphorus. Now the numbers get real small. See that? We go from 1% down to 0.4. It's because everything else are going to be circulating. So potassium and sodium we're going to talk at great length about in the next chapter when we build membrane potential. Chlorine, Cl, chloride, there's not very much of it, and frankly, of all these, chlorine never does anything in the body. I like to joke that it's just around to keep us from sticking to the wall, you know, to keep us electrically neutral, right? Chloride has very little actual physiology happening in the body. And then we get into our trace elements, right, like magnesium, sulfur, iron, iodine, and so on. Very small amounts. All of the heavy metals are important in certain reactions in the body. Iron, you know, is critical for the delivery of oxygen, for the transport of oxygen. Iron is also critical for the capture of energy, so the utilization of ATP and so on. So that is what we are made of. Am I going the right direction? I am. Okay. Who's heard of an enzyme? There you go. Good. Maybe once or twice in one of your many biology or chemistry classes, right? Well, all I'm really interested in you knowing about enzymes in this class is their basic function. Okay, so what does an enzyme do? An enzyme reduces the activation energy of a reaction. So in our top example, we go from reactants to products, and it's true of most reactions, we have to put a bunch of energy in. Well, that energy input, that activation energy, that is going to be your limiting factor on how quickly this reaction occurs. So, in the presence of an enzyme, we see that our activation energy is much, much smaller. So what that means is, compared to this reaction, no enzyme, this reaction with an enzyme is going to occur much more rapidly, and it's going to occur more often, right? So it means that if we want to increase the amount of product that we have available in a cell, if we turn on an enzyme that reduces the activation energy, we're going to increase the amount of the product, right? Because we've turned on an enzyme. That's the notion of regulatability, that we can turn enzymes on or off and therefore affect which reactions are occurring in the body or in a cell at a given time, right? All right. I cover all that stuff? I did. So in cells, the most common enzyme that we talk about, enzymes can technically be anything, but the enzymes, the catalysts that we talk about in physiology are typically proteins. Now, in medicine, which is where you all are headed, we don't always interest ourselves in the details of these reactions. In other words, you learned in biochemistry a lot of mechanisms. How does this molecule in the body become another molecule in the body? We're not going to do a lot of that in this class, okay? I am more interested in you knowing how the human being you can see with your eyeballs functions, not how all their molecules and cells function, okay? Maybe that will make some of you happy, maybe it will make some of you not, but that's the reality of it. We don't get into the chemistry as much as we stay at the physiology. How does it work in organ systems, right? All right, so as important as enzymes are, even more important is water. You've been taught since you were little kids that human beings are mostly water. Well, why? Why did we evolve? Why did life on Earth evolve to have this significant water component? A few reasons. Solubility is one. Water, of course, is known as the universal solvent. It isn't technically universal because there are plenty of things that water won't dissolve, but most things here on planet Earth it will, right? So the fact that water is so has good solvent properties means that all those atoms and molecules that make cells work can interact with each other because they're all in a common solution, right? You can imagine that if we didn't have water, it would be difficult for this part of the body to interact with this part of the body because there's no exchange of material between the two. Whereas in water, because everything is dissolved, we have this medium of water with all kinds of stuff dissolved in it, makes that happen. So solubility. Another reason is reactivity. Water is not inert. I think the way we tend to think about water is that it doesn't do anything, right? We put things into it, we take things out of it, but it doesn't participate. That's not really true. Water participates in a number of key reactions. So, for example, we make water. About 20% of our water metabolism is generated by our own mitochondria, right? We take hydrogen and oxygen from the breakdown of carbohydrates, we combine them together, and we actually form water. So we make some water of our own from sugars, proteins, lipids. That's one example of a reaction. Another example is good old hydrolysis, which you should have learned about in chemistry. That's where we take a molecule that has parts like A connected to B, we add a water to it, and we split A and B, and then we split that water molecule apart, right, into an H that should be an OH, BOH. So that's a hydrolysis, breaking things apart. Hydrolysis is the primary way we digest, so we take in very large organic compounds in our food, and in order to have those compounds be useful to us, we have to break them down into simple sugars, carbohydrates, or fatty acids. Most of that breakdown occurs through hydrolysis, right? So hydrolysis is something we see all the time. Hydrolysis is also used to break up molecules inside the body. So when we take a glycogen molecule, for example, and clip glucose sugars off of it, well, that's an example of a hydrolysis too, okay? Dehydration goes the opposite direction. Dehydration is where we take an A and a B and we combine them together to make an AB, and we get a water out. Now, we like this reaction in the body because guess what? Water we have plenty of and we know how to handle, right? So the byproduct of this combination reaction is something inert and easy to handle. So we like dehydration reactions, right? Okay. Water, you will remember, is polar, okay? That means that its electronegativity is not uniform. That's all that means. And it's because oxygen in the middle loves electrons, right? So it tends to attract the electrons more strongly than the hydrogen ions do. So we end up with a water molecule that has a positive end and a negative end. So we call it polar. This polar property helps water to be the good solvent that it is, right? So here I think I have a picture, yeah. So because water is polar, it has a positive pole and a negative pole. If we put something in water that has electronegativity, for example, a chloride ion, this is sodium chloride, the green is chloride, the purple is sodium. Well, you can see that our chloride ion, it's going to have a shell of negativity, right? Well, that shell of negativity is going to attract the positive end of our water molecules. So what we'll end up with surrounding this chloride, we'll end up with what's called a hydration sphere, where we get a bunch of water molecules stuck all over that chloride. But so you have the negatives from the ion, the positives from the hydrogen or from the water molecule, they kind of cancel each other out. So that makes chloride able to stay in solution, right? Because its negativity is now surrounded by positivity from the water molecules. So it's what makes water such a good solvent. Why can we add so much salt to water and have it continue to dissolve? Because of this, because of the polar nature of water, right? Glucose does the same thing. Now, not for the same reason. You know, chloride has a strong negative charge. Glucose, as a molecule, is neutral, right? But you'll remember from organic chemistry that it is polar, that its negativity and positivity is not uniform. So because, again, of the polar nature of water, in a glucose molecule, we put the water molecules we put the negative end of the water molecules towards the positive sides of the glucose and vice versa, and we get a hydration sphere around that glucose, right? So this is why water is such a good solvent. Now, glucose, is our primary energy source. Because of this solubility, it means that we can have very high concentrations of glucose stored in water, right? That means that we can readily move our metabolic energy around, all right? So a little bit about why solubility is so important. Now, not all molecules are polar, okay? So what happens if you take a nonpolar molecule and you put it in water? Well, you should remember that from organic chemistry too. Here, we have no electronegativity difference. The fats are electrically neutral. So the water has nothing to work with. It has no charge to form a hydration sphere with. So what ends up happening with fats and oils, with the nonpolar compounds, is it separates from water and stays separated. So here you see the fats and the oils floating on the top. There's other examples of this, but it's always the same thing. It's not mixed in. Now, this comes into play when we talk about digesting fats in Unit 3, because our digestive enzymes are water-based, but what we're digesting is nonpolar. You see the problem? So we have to do some fancy tricks to make that happen. Now, also present in this picture, excuse me. Can you tell I haven't talked in a week? All right, also present in this picture are proteins. Proteins are complex molecules that have both positive areas and negative areas. So what we see here is we see hydration spheres or partial hydration spheres with the water molecules, positive end attracted to the negative areas, negative end attracted to the positive areas. So water's polar nature makes it possible for proteins to be much more soluble than they would be otherwise. All right? So water, it's not just, ubiquitous. It actually has these really important characteristics that we depend on. Some other ones, high heat capacity. Water, as you know from the kitchen, right? Water takes a lot of energy to heat up. If you're going to boil water from tap water, you're going to be waiting a little while, right? Because you have to put a ton of energy in to make that happen. Now, we use that characteristic of water as part of our thermal regulation. Because we're mostly water, and because water has a high heat capacity, it takes a lot of energy to raise our temperature or lower our temperature, right? So water's high heat capacity helps us to maintain our body temperature. Water also has some nice tricks to it. Because of its high heat capacity, when water evaporates, it takes a great deal of energy to make that happen, right? So if we put a thin layer of water on our skin, we let air go across it and evaporate that water. That evaporation takes a lot of energy away from it. That's sweating, right? So that's another property we use is water, its evaporative cooling potential means that we can maintain our body temperature. This is a tricky thing we can do. We have 98.6 degrees. We can go outside on a day like yesterday, right, when it's 100 degrees and maintain our temperature. That shouldn't be possible, right? It's only possible because of this evaporative quality of water. If we didn't evaporate water, we would also be 100 degrees out there, okay? So key aspect, large amounts of energy required to change the temperature and easy to evaporate the water off. The vapor pressure. Vapor pressure of water is quite low. That means we tend to hold on to it. Think about the difference between, you've all seen acetone, okay, in the chemistry lab, yes? So acetone, you leave it on the counter overnight. Water, you leave it on the counter overnight. What's the difference? Acetone will evaporate. It'll be almost gone by the time you get there in the morning, and the lab will stink, right? Why? So that's vapor pressure. Acetone has a very low vapor pressure. It wants to go into gas. Water, low vapor pressure, doesn't want to go into gas. So it means that we don't have to keep replacing water we're evaporating all the time, right? Because while it does evaporate, it takes some effort to do so. So low vapor pressure. Lubrication. Water's polar nature makes it slippery. The hydrogen molecules, the polar areas, the negative areas and the positive areas, they tend to slide past each other in water. So it means that a small amount of water between two surfaces greatly reduces the friction between those two surfaces. We use this all the time. So every time you take a breath, the lungs have to slide inside the chest. Well, in order for that to occur very easily and not get bound up, we need to have lubrication. So what do we have? Inside the pleural space, we have a small amount of slippery fluid that's mostly water. Okay? So lubrication is a major player of what water is about too. Keeps things moving and sliding nice and easily. All right, so let's get your Kahoots out. All right. Let's see what you remember from chemistry. 3, 2, 1. All right, movement down the concentration gradient. What do we call that? All right, very good. That's diffusion. Osmosis is also movement down the concentration gradient. You'd get me on a test on that one. And it's spelled wrong. Did everybody catch that? Osmosis? Yeah. So that's diffusion. Now, Kahoots. One thing I want you to know, Kahoot questions are about learning today, all right? So what that means is Kahoot questions are far easier than my test questions are. Okay, I don't want you to have too much of a false sense of security. All right, because these are like basic knowledge questions where my test questions are going to be more putting the pieces together. All right, and we'll talk more about that before we get to a test, but I just didn't want you to think they'll be this easy. All right, Kendall's leading. To have diffusion, you must first have what? All right, do read all the answers, okay? It's all of those. So yes, a difference in concentration is necessary for diffusion. That's what we call a diffusion gradient, okay? A diffusion gradient is a difference in concentration. And then the ability for the interesting molecule to move. There can't be a barrier, all right? So for example, in the part of the kidney and part of the nephron, there's an area where the concentration gradient is huge, but water doesn't move. And it's because it's impermeable to water at that point. So there, you do still, the molecule of interest has to be able to move. Oh, some changes. The wise owl. All right. Osmosis. Did I spell it right? I did. All right, best answer there is, you're going to hear that term a lot, best answer, is the blue one. Movement of water from high solute concentration to low solute concentration. Now, why not the result of a semi-permeable membrane? It is if there's a concentration gradient difference, right? But a semi-permeable membrane in a fluid with no difference won't cause any osmosis to happen, okay? So best answer there is the blue one. Oh, purple tiger coming up. 2 more, then we'll go on. All right, what does an enzyme do? Wow, that was fast. Good. That's all of those. Makes reactions happen faster, more often, and with less energy. It's why they're so important in the body. That and the fact that you can turn them on and off, right? Think about it. If we couldn't turn enzymes on or off, we would be the same all the time. Our physiology would always be the same, kind of like a computer, right? So the fact that we can be different depending on our needs is mostly because we can turn enzymes on and off. All right, I think this is the last question for now. All right, a polar molecule has which of these? All right, very good. That's a positive end and a negative end. That's what you need. All right, let's see who the winner for this round is. Nicole is number 3. Purple tiger. Katie's the winner. All right, good work. All right, we will leave that. Go back to that. Okay. Okay, so moving away from water and the basics and into the molecules of the body. So first off, electrolytes. Electrolytes are simple little molecules that have big functions. Okay, you know, the organic compounds get a lot of attention in the body because they create structure and they are responsible for the unique life processes. These electrolytes are present all over the planet, and they're present in us too, and they play a role there as well. So the top two are the major players, sodium chloride and potassium chloride. Those are your classic electrolytes. Sodium is the predominant cation in the extracellular fluid, okay? Potassium is the predominant cation in the intracellular fluid. That difference, that sodium and potassium have a constant concentration gradient across cells, is the foundation of membrane potential that we're going to talk about in the next chapter. All right? So that's those two. Calcium phosphate is one of our sources of calcium. That's calcium phosphate is the primary form in the bone, right? Calcium phosphate. Sodium bicarbonate. Bicarbonate in particular, it plays a major role in our acid-base balance. Okay, we have a whole lecture pretty much on bicarbonate later on. And then magnesium, the other phosphate, the hydrogen phosphate, and sodium sulfate are less important. Okay, but the key to the electrolytes, an electrolyte is defined as a substance that diffuses into ions. Okay, so if we're calling it an electrolyte, it means that you've got positive and negative charges in water somewhere. Okay? All right, so next up then is the carbohydrates. You know, you should remember carbohydrates vividly, I would think, from biochemistry. Just as a refresher, they have a 1-to-1 ratio of carbon, hydrogen, and oxygen. That's one of their claims to fame, right? 1-to-1. Carbohydrates are our preferred source of fuel in the body. So much so that we turn other things into carbohydrates when we go to create energy. So for example, we turn proteins into carbohydrates before we break them down to form ATP. Okay? So preferred source of fuel, many other functions. I'm trying to get the volume right here. Many other functions as well. Carbohydrates play a role in structure as well as function. All right, so like cell membranes have a coating of carbohydrate called glycocalyx. So even though we talk a lot about carbohydrates as fuel sources, they do other things. That's what I'm getting at here. In terms of the fuel source molecules, monosaccharides, glucose, fructose, galactose, those are the common ones. Zooming in a little, glucose, of course, 6-carbon ring, sometimes represented as a chain. You know, you can unfold the ring, so to speak, bless you. But why is that important? Rings are very stable. Rings are a way of sharing the reactivity of a bond across many different atoms. So we get a lot of energy in a ring. And what that means is when we break this ring into its component carbohydrate or component carbon pieces and form carbon dioxide, we end up releasing a lot of energy. So glucose is our primary energy source for a couple of reasons. Number one, it's rapid. Okay, we can metabolize glucose more rapidly than any other substance, right? Number two, it contains a fair bit of energy, not nearly as much energy as a fat, but we can get at it a lot faster, right? And glucose is readily storable in the body. Number one, it can exist in relatively high concentrations as glucose, and then we can also store, we can attach glucose molecules together to form starches and store even more that way. All right, so plants tend to make disaccharides. Now, disaccharides are two simple sugars stuck together with a simple hydrolysis. So table sugar, sucrose, is made of a fructose saccharide and a glucose saccharide attached with an oxygen, right? And we can separate those very easily through hydrolysis like we talked about earlier. So in our digestion process, we have to break the sucrose into glucose and fructose before it gets absorbed into the body, right? And then the glucose and fructose are what get fed into the mitochondria to produce ATP. Everybody get that? Okay. Now, what about the polysaccharides? Well, in animals and in plants too, we not only make disaccharides, but we make polysaccharides. All right, so think about this for a minute. You've got lots and lots of cells in the body. All of those cells need glucose as a fuel source. Well, if we took all of the glucose that the cells needed and just shipped it around the body as glucose, right, you could imagine we'd have a very high glucose concentration, okay, which isn't really good for us. That's diabetes, right, when our glucose concentration is too high. All right, so if we have all these cells that need glucose, but we don't want our glucose levels to be super high, well, what do? Well, what we do is this trick. We take our glucose molecules and we covalently attach them together to form what's called a starch. Okay, a starch is an example of a polysaccharide. A polysaccharide is a bunch of glucose molecules that have been chained together covalently. Now, why is that useful? It means we can store all these glucose molecules, but not in solution. Do you see that? So our blood glucose level is still normal, even though we've got a bunch of glucose sitting around in the liver. Okay? Our starch that we make is called glycogen. Glycogen is long branched chains of glucose molecules that the liver has put together. Now, the beauty of this system is it is very easy and cheap metabolically to add glucose molecules to this or to break glucose molecules off of this. So, for example, it's 6:30 at night. You've just had a great meal at the Olive Garden, right? Lots of pasta, plenty of carbohydrates circulating. So what happens? In the liver, all those glucose molecules are going to get attached to other glucose molecules, and you're going to load up on glycogen. You're going to form a bunch of glycogen. Now, skip ahead, 6:30 in the morning. You haven't had anything to eat since Olive Garden 12 hours ago. So now what happens? All those stored up glucose molecules get clipped away and released into the blood so that your blood sugar stays normal even though your intake went way up and way down. Do you see how that works? So this is polysaccharide glycogen. Still a carbohydrate. Okay, so just like those are carbohydrates, glycogen is also a carbohydrate. It's just a much, much bigger one, okay? And glycogen is not very, this is glycogen is not very soluble. So it falls out of solution as granules. So when you look in the liver at midnight, right, you'll see a lot of cells that have glycogen granules, stored up glucose, ready for the fall in blood sugar that will inevitably happen at 4, 5, 6 in the morning. Right? When you haven't had anything to eat for 8 hours. Okay. All right, that's carbohydrates. How about lipids? Lipids also contain carbon, hydrogen, and oxygen, but the ratios are different, right? We don't have the 1-to-1. We just have a 1-to-2 ratio of carbons to hydrogens with very little oxygen. All right? Now, lipids typically are mostly carbon and hydrogen, tiny bit of oxygen, but they can have other players. They can have phosphorus, nitrogen, sulfur, and so on. And we'll talk about some of the lipids here in a minute. Lipids are insoluble in water, which causes a problem in the body. You know, if we need lipids to survive, but lipids don't mix in water, how do we move lipids around when our blood is mostly water? Well, we have a system for that. We have proteins that we put lipids inside of called lipoproteins and move them around that way. But we still have to have special transport systems. Lipids serve as energy reserves in the body because they're dense. Okay, fats store about twice as much energy per gram as carbohydrates do. So it means if what we want to do is store energy, fat's the way to do that. All right, and that's what triglycerides are for. Now, when you get into your full body donor in the anatomy lab, one of the things that you're going to see, well, a lot of, is fat, right? Anybody ever seen the donors before? Did you do that yesterday? No? Trust me, you're going to see a lot of fat this semester, and it's going to be in that lab, right? Okay, so why? Why is there so much fat in our donors? Because anywhere that the body needs to insulate, pad, or lubricate, fat's why we do that, all right? So, for example, in the heart, okay, you're going to open up the chest cavity at some point. You're going to open up the pericardial sac. You're going to pull the heart out, and it's going to be a lot fattier than you think. Now, why? Does that mean that they were sick, they had some fatty heart? No, it just means wherever there's an empty space, the body's going to pack it with fat, right? Because unlike your box from Amazon Prime, there can't be any empty space, right? It has to be complete, no spaces. So wherever there's spaces in the body, we usually fill those with fat because it's a nice, soft, lubricating compound. Fats are used as the base molecule for many hormones, estrogen, progesterone, testosterone, to name a few, because they can pass through cell membranes. So one of the things we'll talk about in the next unit when we talk about cell membranes is that cells, the membrane is made of lipid, right? It's phospholipids. So what can pass through lipids? Water or fats? Fats can pass through, right? So the reason that our hormones are fat-based is because by being that way, they can pass right through the membrane and they don't need a transporter then, right? So more on that later. And then finally, the lipids form essential structural components of cells, very specifically the cell membrane. The cell membrane is made of phospholipids, and phospholipids are a specific kind of lipid that have a polar end and a non-polar end, all right? So this is our dear friend, the fatty acid. This is the most simple kind of fat. So you can see that it's a long chain of carbons with hydrogens balancing out. And then only at the one end do we really have anything interesting. And we have 2 oxygen molecules and a donatable hydrogen ion. So this little guy right here off on its lonesome, when you put that in water, that little guy comes off. That's why we call it a fatty acid, because it's a proton donor, right? And what's left then, in this case, would be a lauric acid with a negative charge on one end. Everybody see that? So fatty acids are proton donors. Fats come in 2 varieties, saturated and unsaturated. That refers to how many double bonds they have, all right? So like a polyunsaturated fat is a fat that has multiple double bonds. Poly is many, unsaturated is double bonds. A saturated fat has no double bonds, all right? So the saturated version is the simpler version, but the unsaturated version is a little easier to metabolize. So while research has gone in both directions, we still typically say that saturated fats are a bit less healthy than the unsaturated fats are, right? One of the things that's significantly different about these two is melting temperature of all things, right? So like when they go to make margarine, for example, they have to hydrogenate. They have to change the saturated and unsaturated ratio in order to make it solid at room temperature. All right. Glycerides. A glyceride is a fatty acid, so one of those, okay? It's a fatty acid that we've attached to a glycerol backbone. All right, so let's do a little organic chemistry, right? So this is our glycerol over here. This is 3 fatty acids. 2 are saturated, 1's unsaturated, okay? Now, on our business end, on our proton, whoops, wrong way. On our proton donor end, right, so we're on that end, what we've done here is we've taken that end and we've attached it to the OH on the glycerol. So what we end up with is 3 fatty acids covalently bound to a glycerol molecule. This is something you've heard of. This is called a triglyceride, all right? Triglycerides are the primary fat that our body produces both for transport and for storage, all right? So when you're looking down into your donors, the yellow stuff that you see is all triglyceride, right? So it's in adipose tissue, it's wherever fat is found. Now, the nice thing about triglycerides is this is really a storage form. When we go to use this molecule, we're going to go backwards. We're going to clip off this fatty acid, feed it to the mitochondria. Clip off this fatty acid, feed it to the mitochondria, and so on until we don't need any energy anymore, right? So much more on that when we get to our metabolics and energetics chapter. All right, another kind of lipid that's important is the icosanoid. These are molecules that play a major role in cell-to-cell signaling, all right? You all are in an immunology class, correct? You're going to learn about the outcomes of these. So, for example, the leukotrienes and the prostaglandins are cellular messengers that play a huge role in the immune system. The molecule they originate from is an icosanoid. The icosanoid is a carbon ring with 2 short fatty acids attached, okay? Now, I get to say something happy to you right now, and that is you don't have to memorize any clinical or chemical formulas from my class, okay? I show you the pictures because I want you to understand what these molecules look like so that you understand their properties. This is not a chemistry class. There's one equation that I'm going to be interested in you knowing, and I'll tell you when we get there, okay? But it's the bicarbonate equation of pH balance. But otherwise, please don't feel like you need to identify, name, or otherwise reconstruct this molecule. I want you to know that an icosanoid is a lipid that is used to form leukotrienes and prostaglandins. Get it? Okay. We're getting to know each other, right? So you got to know what I'm expecting of you and vice versa. And please ask questions if you're not sure. Okay, you've all heard this term, steroids, right? Well, what is a steroid? A steroid is just a compound or a molecule that has a hormone-like effect in the body. And steroid, that word, describes a molecule that has this backbone, this complex 4-joined ring structure, all right? So cholesterol is what we ingest. This is where it comes from. Cholesterol comes from animal products, right? So meat, for example. We can also produce our own cholesterol if you're a strict vegetarian. But the cholesterol is metabolized to form hormones like estrogen and testosterone. Progesterone is also on this list. There's a long list. Short version, this connected ring structure gets modified to form hormones. Why? Because this structure can pass right through cell membranes, so it does not need a receptor. You can imagine that some hormones, we don't want to have the cell to need a receptor to respond to it, right? Because, like the steroid hormone, so we'll pick on estrogen, for example. Estrogen affects every cell in the body. The hormones for estrogen's effects are found in the nucleus of cells. So how do you get from the outside world into the nucleus? Well, you got to get through two cell membranes, right? A nuclear membrane and a cell membrane. Well, how are you going to do that? By being a molecule that's nonpolar. That's how. All right? So we'll pick up the story of these hormones at the end of the semester when we get to endocrine, right? So for today, it's just know the properties, that they're lipid-based, they can pass right through cell membranes. All right, so one of the most prevalent lipids in the body is this guy. This is a phospholipid, all right? So we have our fatty acid tails, okay? We have a glycerol backbone right here. Then we have our fancies. We got a phosphate group and a non-lipid group up at the top. That might be a carbohydrate, might be other things. Okay, so phospholipids have the phosphate group. Glycolipids have carbohydrate groups. Now, what makes phospholipids so important, not only in our body, but in terms of life on Earth, right? Cells would not exist without phospholipids. What makes phospholipids important is they have a hydrophilic head and hydrophobic tails. They're bipolar, right? And I don't just mean they have a plus and minus. I mean they have two different personalities. They have a hydrophilic personality and a hydrophobic personality. Okay, so why is that important? Because it means they can arrange themselves into a lipid bilayer. All right, so if you've got a bunch of molecules who have one end that loves water and the other end that hates water, well, how are they going to end up orienting? There's two things that might happen. This is one where all of the tails, the hydrophobic tails, are pointed together and all the hydrophilic heads are pointed out. Now, the other way that this can happen, I don't have my fancy drawing thing yet, but the other way that this can happen is hydrophilic heads pointing out, hydrophobic tails pointing in. Man, that's a terrible marker. Hold on. There we go. Okay, so these are our two approaches. One is to make a ball. This is called a micelle, all right? The other is to make a bilayer. Do you see how the hydrophobic parts are pointed together and the hydrophilic parts are pointed towards the water? See that? Well, we can take this lipid bilayer, curl it around. Oh, I don't have a picture of that. Curl it around and make a cell out of it, right? So we can have two layers. More on this when we get to the membrane. These two layers then are going to go all the way around and make a sphere. The inside of that sphere is the inside of the cell. The outside of the sphere is the outside of the cell. Get that? Okay, more on that later. More on phospholipids later, too. Whoops. Okay. I'm going backwards. Phospholipids, proteins, there we go. All right, so the last, well, I don't know if it's the last. It's not the last, but proteins. So what do proteins do in the body? Proteins do lots of different things. The keys: support, movement, transport. Support is the structure of the body, is not entirely protein, but without protein, we wouldn't have the structure of bone, muscle, so the shape of things. Why are we the shape, and why are organs the shape they are? Protein. Movement, muscle, right? Muscle is a protein organ. And we're going to talk at length about how muscle works probably later this week. So we'll talk about how protein makes movement. Protein makes transport happen at the cell membrane and in the blood. So it's hemoglobin is a protein that's responsible for carrying oxygen around the body. And there are numerous other examples of transport moving things around. I told you about one, the glycolipids, or the the lipoproteins that carry lipids around the body. Buffering is the concept of controlling pH change. Okay, we have a number of molecules in the body that resist the change in pH. We have a whole chapter on that too. Proteins are the major intracellular buffer. So proteins play a role in maintaining the body's pH. Metabolic regulation, we talked about this already today. Enzymes. Many proteins form or play the role of enzymes in the body. And regulatable enzymes, so things that can be turned on and off and therefore changing cell function. Coordination and control. Proteins are the switches that control cellular behavior. If you think about it, every given cell in the body has to do some things sometimes and other things other times, right? So how does the cell know when to switch activities or behavior? Well, hormone system primarily, and the nervous system, and protein plays a role there. And then finally, defense. You're going to learn all about the defense of the body and immunology, but one of the things you're going to learn is that proteins play a major role here. So for example, the membrane attack complex of the complement system is an example of proteins playing defense. Another role there is antibodies, right? Antibodies are small proteins that also play an important role. All right, so if proteins are all this important, what are they made of? Well, this is the schematic of a protein's building block, which is an amino acid. So we have an amino group on one end, carboxylic acid on the other. We have an R group. This is what defines the amino acid as what it is, right? So each R group is different. And then we have our lonely H up there. So all of the proteins are made up of long chains of amino acids. Does that sound familiar to anybody? I hope so. Otherwise, you miss a lot of biochem, right? But proteins are long chains of amino acids. There's really only 20, is that right? 20 different amino acids? 22, yeah. I don't know why I only have 20 up there. But anyway, you have these different amino acids that we put together in different ways to form the different proteins. Each of those different amino acids, the R is a little bit different. So just to refresh your memory of how this works, we take two amino acids, we do a dehydration synthesis, and we link them together in what's known as a peptide bond, right? We do this a few thousand times. So we end up with this long chain of amino acids, right? And where do we get the blueprint for the long chain of amino acids? DNA. Very good. So you paid attention that day. That's very good. Typical protein, 1,000, can be as complex as 100,000. These can get very, very long. The primary sequence, or the linear chain, folds up on itself. Because of the chemical properties of the R groups, proteins are forced into a transformational change, okay? So proteins are 3D structures. They're not long chains. They're 3D structures made of long chains. So some of the themes in these structures, we can take a chain and we can do two things with it. We can coil it up. That's what we call an alpha helix. Or we can make a sheet out of it. We can go like this, like you're painting mowing the grass, right? That's called a pleated sheet. Now, we can take those basic three-dimensional structures and then combine them into secondary structures, which is a complex protein shape, and then tertiary structures, which are complex shapes wrapped around themselves, and then quaternary structures are when you get more than one protein unit that's stuck together. Okay? Again, that should all be review, I hope. All right, so some of those proteins that look like that, right, behave as enzymes. Enzymes have a few key characteristics that make them important. We talked about the main one, which is lowering activation energy. But we also have these two other characteristics I want you to know. One is saturability or saturatability, depending on which dictionary you want to look at. But saturatability is the notion that each enzyme is performing an enzymatic step. So at some point, more reactants isn't going to make your reaction happen any faster. Okay, so here, I'll just give you an example. So you've got a beaker that's got a ton of glucose in solution in it, right? Into that beaker, you put a fixed amount of enzyme that breaks down glucose, right? The concept of saturatability says this, that reaction that's catalyzed by that enzyme is only going to be as fast as there is enzyme available. In other words, it's not going to go faster if you add more glucose, right? Because the rate of reaction is determined by the number of enzyme molecules, not by the amount of glucose that's present. Does that make sense? So what it means, saturatability, means that at some point all of the enzymes are working as fast as they can. So further changes in the concentration gradient doesn't actually make anything different happen. That's the notion of saturatability. All right? The other idea is regulatability or regulation. This we've been talking about already. Enzymes can be turned on or off. So that means that at any given time, a cell can activate or deactivate whole metabolic pathways by activating or deactivating enzymes. And when we talk about regulation in this class, that's one of the ways we're going to talk about it, is that signal affects a protein, that protein affects cellular metabolism, which changes what that cell does, right? So that's regulation. We can turn enzymes on or off through external signals. All right. So a little bit more about enzyme function. You know, this is a classic example of how does an enzyme work. Well, one of the ways is that an enzyme will co-locate or put together in space the two substrates that we want to have react. Okay? So one of the limits on how quickly a reaction can occur is if molecule A can get access to molecule B. Well, if we have an enzyme that puts molecule A right next to molecule B, well, it means that this reaction to form a product can happen much more easily, right? Do you see that? Than if these two things were just wandering around trying to find each other. So this is one example of how an enzyme works. Some key characteristics of enzymes: they're reusable, so one enzyme can catalyze millions of reactions, but they are delicate. That means you can break an enzyme. We do it all the time when we cook, okay? So if you take a protein and you heat it up, you change its shape. If you change its shape, it can't function in the same way anymore. We call that denaturization, or denaturing the protein. So when a enzyme's shape changes, it is denatured and then no longer functions. So some of the things that change shape, pH will change the shape of an enzyme. We're very sensitive to pH. Our normal pH is 7.4. If I dropped you right now in a hurry to 7, only 0.4 difference, we'd be putting you in the ICU. Okay, so we're very sensitive to pH. Temperature, same thing. You know, yes, we get a fever, right? But after about 110 degrees, our enzymes don't work so well. Okay, so you're talking about a fatal high temperature at that point. So just two examples of things that can affect the structure of the enzyme and therefore its function. All right, a little bit about adenosine triphosphate. This is the energy dollar. Hopefully you've learned lots about this in the past, but this is how I talk about it, is as the energy dollar. I call it that because, you know, think about our way we interact with dollars. Well, not right now because you all are unemployed, right? but someday you won't be, right? So how do we work with money? We do a task, and somebody gives us dollars for that task. Now, the beauty of dollars is we can take those dollars and we can go do whatever we want with them, right? In other words, how we earned the money has nothing to do with how we spend the money. Well, in the cell, it's that same way. The dollar is ATP. So the cell has a few ways of making the dollar. Mostly that's happening in the mitochondria. You're familiar with that already. So we have a few ways to make a lot of ATP, and then we have a lot of ways to spend that ATP to do different things, right? So that sort of is the nature. We make the ATP, and then we spend it by taking ATP and breaking it into ADP and P. Now, when we break that bond, we release energy. That energy can then be used to power other reactions. So, for example, if we're moving a molecule up its concentration gradient from an area of low concentration to an area of high concentration, right? So opposite the normal, we have to use energy to do that. So we might have a pump that splits an ATP into an ADP and P, then uses that energy to push a molecule from a low concentration area to a high concentration area, right? This happens in the kidney all the time, for example. So that's adenosine triphosphate. Now, the most common way is ATP to ADP and back, right? But we can have the other forms. We can have adenosine monophosphate, diphosphate, or triphosphate. It goes in order of energy contained. The ATP has the most, ADP next least, and then AMP the least, all right? So we usually go ADP and ATP and back again. So here's what that molecule looks like. We get an adenosine and then 3 phosphates that we either cleave or add. Now, why phosphates? Phosphate to phosphate bonds are high energy, but relatively easy to break. So it makes our dollar easy to spend, which is what our body wants. All right, so like I said, this is the most common. We take ADP and energy and add it to P. So when we form ATP, like in the mitochondria, the energy is coming from the breakdown of carbohydrate, right? When we're spending ATP, we're going from ADP to P plus ADP, right? Now, the beauty is the adenosine gets reused. So same adenosine might be an ADP and an ATP thousands of times, right? It might just go round and round and round. Okay. All right, our last topic of today is nucleic acids. Nucleic acids, while proteins form structure and participate in a lot of metabolism, nucleic acids, their role is really to store and transmit information, specifically information for how to make proteins, right? So we'll talk a little bit more later on about the interaction of DNA, RNA, and proteins. But for now, just know that nucleic acids are made of these building blocks called nucleotides. Nucleotides are strung together then to form DNA and RNA, also using a dehydration reaction similar to what we saw in amino acids and proteins. And then these nucleic acids, the long strings of them, are going to form the template for proteins and for protein synthesis then. So in the end, you end up with this structure. Now, this is not a molecular biology class. I am not going to be asking you to match up C's and G's and A's and T's, okay? If maybe you've done that, maybe you haven't. I promise you it won't come up in medicine, okay? Because it doesn't. But I do want you to know the conceptual stuff, right? That RNA is used as a blueprint, right? So RNA makes kind of a carbon copy of the DNA. The RNA then leaves the nucleus, and that template is used as the blueprint then to form a protein that then goes on and does something, okay? All right, we have a few minutes to do another Kahoot. I made it through the first one. Molecules. All right. Disaccharide, glucose, fructose, and starch. What do they all have in common? Hey, very good. Yes, those are all carbohydrates, right? Disaccharide is two or more simple sugars. Glucose and fructose are both simple sugars. And then a starch is just a long branched or unbranched chain of glucose molecules. All right, boom. Molecules that have long carbon-hydrogen tails are which of those? All right, good. And not all lipids have long carbon-hydrogen tails, because remember we saw a few of those, like cholesterol and progesterone, estrogen, some of the icosanoids. But the vast majority of the lipids found in the body are of the fatty acid type with the long carbon-hydrogen tails. All right, Caroline's the winner. All right, what makes amino acids different from each other? Man, you just made some biochemistry teachers very proud. Or was that organic? Both? All right, last one. A fixed amount of enzyme speeds up a reaction. Adding more reactants doesn't speed things up. Why? Yeah, that's saturability or saturation. The idea here is when all the enzymes are busy, more reactants is not going to make things go faster because you need more machines is what you need, right? Okay. All right, and we'll find out who the winner is and then call it a day. Logan is in third. Cass is in two. And the winner is Carolyn. Where is Carolyn? No, Carolyn? Hi. Yeah, okay. All right, that's it, folks.